Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, a division of Twist Bioscience, I'd like to welcome you to discover antibody candidates against a panel of hard-to-drug GPCR targets, a synthetic antibody library case study. My name is Elizabeth Lamb, and I'll be your host and moderator for today's event. Now I'd like to introduce our presenter for today. He is Dr. Aaron K. Sato, PhD, Chief Scientific Officer, Biopharma, with Twist Biopharma, a division of Twist Bioscience. Welcome, Dr. Sato. The presenter ball is yours.
Thank you, Elizabeth. Again, my name is Aaron Sato. I'm the CSO of the Biopharma vertical at Twist Bioscience. It's my pleasure to talk to you today about a case study where we've used one of our antibody phage display libraries that's focused on drugging hard-to-drug GPCR targets using our synthetic antibody phage display libraries. So again, I always say the best companies out there really truly understand the one thing that they're really good at. So for Twist, that's our ability to print DNA. So shown here on this slide is a silicon chip. It's about the same size as a 96 wall plate. And on this chip, we can actually print individual oligos. You can actually print up to 1 million different oligos on this single chip. And those oligos can be up to 300 base pairs in length. So each one of those individual oligos has an explicit sequence. And so this is a fantastic platform to make large pools of individual oligos. And of course, we use this platform to make all kinds of products that I won't talk about today. But again, clonal genes is one of them. And in particular, we can use pools of oligos to make high quality DNA libraries, which is key for the case study that I'll talk about today. So how do we do that? So basically what we can do is we can again take those large pools of oligos, and if you think about an antibody domain, so again either a heavy chain or a light chain domain, it's basically a collection of 3 different CDR loops in a single germline framework, for example. We can basically make individual pools of oligos that encode for diversity and then stitch those pools of oligos together to make a hypervariable, either heavy chain or light chain domain. So shown here is just one example where we might, for example, have a heavy chain domain library that we're trying to make, where we might make, say, for example, 1,000 oligos that encode for the diversity in CDR1, another 1,000 for CDR2, and maybe 10,000 or more for CDR3. So the reason why making libraries in this way, in my mind, is really game-changing is that, for example, when I synthesize those oligos in those pools, I can make sure that they exactly match the human repertoire. So the resulting antibodies that come out of the library look a lot like you would find in any human antibody derived from a person. I also can remove sequences up front from the synthesis that might lead to issues with isomerization, cleavage, deamidation, or glycosylation, for example. And then finally, and the third advantage, which is actually key to the GPCR library case study that I'll talk about today, is that we actually can encode for specific motifs in those sequences if we choose to. So in Twist Biopharma, we're focused on several strategic areas. I'm going to focus almost exclusively on the second one shown on the slide, which is that we can make human antibody phage display libraries that are focused on difficult to drug classes of targets. And the first class of targets that we've gone after is the most widely validated but difficult to drug class of targets, G protein coupled receptors. So we now have 2 libraries. I'll talk about one of them in depth today, which is our motif guided GPCR antibody library. We've also created a whole series of other antibody phage display libraries that are more general use, both Fab, single chain, and VHH libraries, which I won't talk about today, but certainly please follow up with me if you're interested in those. And then we also have a platform for doing antibody optimization. So if we find an initial lead from our libraries, we can always make it better through our optimization platform. That I also can provide you information later if you're interested. And then finally, the fourth area that we're focused on is trying to extend the Twist SynBio offering. Again, Twist is a major player in the space to synthesize clonal genes, but we're trying to extend that into antibody production. So someday in the not too distant future, you'll not only be able to order an antibody gene, but also the antibody that's encoded from that gene from Twist. So again, our advantage that we see in this space is that we can make precisely defined libraries using the library platform I just talked about to make unprecedented, very high quality antibody phage display libraries that are devoid of any liability issues and match the natural human repertoire, and also potentially contain sequence motifs, which is, again, as I said, is really key to the GPCR library that I'll talk about today. So the vision for the Twist Biopharma vertical is, because we sit on top of this fantastic DNA company that can make libraries really fast and of high quality, that we see that eventually someday we will potentially have a library of libraries. One way to potentially address the limitation in sequence space of a lot of libraries that we generate, which in the phage display realm is typically around 10 billion antibodies in a library, is just to make more libraries. So if I can someday have say 100 libraries, each one having a diversity of 10 to the 10th, then I would basically have 10 to the 12th different antibodies that I can pan against any individual target. So that's the vision, and we've already created upwards of over 10 libraries already, and by the end of the year, we'll probably potentially even have upwards of 30 or 40 libraries that we can offer to our partners. So again, I probably don't need to convince you that G protein coupled receptors are an important class of targets. If you actually look at all the targets that are drugged by either small molecules or antibodies today, roughly about half of them are G protein coupled receptors, and they cover a broad range of indications. So not just oncology, but there's metabolic indications, inflammation, respiratory, a whole gamut of different diseases. So very well validated class of targets, but the difficulty with this class of targets is because of their integral membrane nature, it's been with very little exposed surface area, it's been difficult to find antibodies that bind these particular classes of targets, as you may know. But they have been drugged quite successfully with small molecules. So but in the industry, people definitely want to potentially move towards antibodies because they have greater specificity and also longer half-life. And to date, there has been some successes. There's actually 2 approved anti-GPCR antibodies on the market today. But certainly, I think folks would really value and appreciate a turnkey library approach to potentially drugging any additional target, and potentially unlocking this really valuable class of targets. And so our approach is, can we create a fully human synthetic antibody phage display library where we don't need to immunize an animal, and we can basically pan and screen that library against any GPCR we wish to potentially find antibodies not only bind those receptors, but also have specific function. So how did we do that? So we took a lot of learnings from this paper back in 2014 by Liu et al. And what the authors had done in this paper is they took a known crystal structure of CXCR4 bound to a loop peptide antagonist called CBX15. And they hypothesized, could they take this loop peptide and basically graft it onto an antibody scaffold to enable that antibody to now bind to and potentially inhibit CXCR4. And so the antibody scaffold they chose was this bovine antibody, which bovine antibodies, as you may know, have a very long, extended heavy chain CDR3 loop. And oftentimes for some of the longest ones, they actually have this knotin structure at the top. So what the authors did is they took off the knotin from the bovine antibody and actually grafted on the CBX15 loop peptide. And what they found is the antibody then bound to CXCR4 transfected cells, shown on the right, and also retained the antagonist function of the original peptide. So their hypothesis actually rang true, that they actually could create a GPCR antibody that had binding and function. So from this, we wanted to create a library that followed the same rules. But rather than just have one motif derived from one particular peptide, could we create a fully human antibody scaffold, not a bovine one, where we had a very large oligo pool that encoded for not just one motif, but maybe hundreds of thousands of different motifs of all the things that are known to bind to GPCRs. And I'll get into the source of that content in a second. But that's the framework for building the library that I'll talk about today. So where did all those motifs come from? So of course, many GPCRs bind chemokines, small proteins that have a specific fold. It is known in literature which parts of these chemokines are central to binding to G protein coupled receptors. And so What we did is we took specific pieces of those chemokines that have that known interaction and used that in the library. The second is, of course, a lot of GPCRs actually do bind peptides. So we took the central core of those helical loop peptides that are known to bind to specific GPCRs and encoded that into our library. We also took some information from GPCRs that are known to interact with themselves. It is known that the N-terminal domains in many GPCRs actually interact with their second extracellular loop. And so we took some motif content from the N-terminal domains of many GPCRs. And then finally, there are a fair number of anti-GPCR antibodies out there, and we've taken some heavy chain CDR3 motif content from those and also incorporated that into our library. So again, the idea is we synthesize this very large oligo pool of over 100,000 oligos that encode for all these motifs, and now we're going to attempt to graft them into an antibody heavy chain CDR3 loop to enable that library to go after any GPCR we wish. But before we could do that, we had to create that antibody scaffold that would accept those motifs. And also we wanted to create a very long heavy chain CDR3, much like the bovine antibody example that I showed before, to enable that loop to insert itself into the GPCR and have function and potentially block or either activate or block that particular GPCR. So what we did is we scoured our databases for all the known long heavy chain CDR3s that we could find. And the idea here was basically to take the N-terminal piece as well as the C-terminal piece of each of these long heavy chain CDR3s, and then basically take out the middle and insert these motifs that we've now gathered. And so what we wanted to do is figure out which long heavy chain CDR3s could support this motif grafted scaffold. And also we wanted to understand which heavy and light chains would also support that kind of structure. So we basically spent an entire year looking at different heavy and light chain combinations, as well as different long heavy chain CDR3 stems that could accept a foreign motif sequence. And as a surrogate for our motifs, before we put this large oligo pool diversity into it, we actually used a flag tag as a surrogate for that. So that's what's shown on the next slide. So what we did is we basically, as I said, looked at different heavy and light chain combinations and long heavy chain CDR3 stems in the context of using that flag tag to look at the displayability of different heavy and light chain scaffolds with different long heavy chain CDR3s. And at the end of the day, we ended up settling on 2 heavy and 4 light chain frameworks and 5 different heavy chain CDR3 stems that could support display of a foreign motif sequence in the context of a phage display library. And so at that point, we felt ready to then make our final library, where we inserted an oligo pool of over 100,000 different oligos that encode for those specific GPCR motifs into our final library. The library is, again, a phage display library. It's fully human, and it's an SCFE-based library. And we transformed it on the level of 10 to the 10th, so it has a overall diversity of 10 billion different anti-GPCR antibodies in it. So as proof of concept for this library, and we basically created a whole series of stable cell lines that encode for specific GPCRs. I'll talk about 3 of them today. The 3 targets I'll talk about today in my case study are GLP-1 receptor, CXCR5, and CRTH2. For each of these targets, we basically created this hybrid construct for the GPCR where we encoded a flag tag on the N-terminus of the GPCR to look at cell surface expression when we created stable CHO lines, and then also a GFP tag on the C-terminus to track total expression. And so as we were making our stable cell lines to pan and screen against, we were able to look at cell surface as well as total expression in our stable CHO lines. And shown here is some example data where you see high level of cell surface expression for, in this case, CXCR5. So in terms of our workflow for discovering antibodies to GPCRs utilizing this library, again, we have our highly diverse GPCR-focused SCFE phage display library with a diversity of greater than 10 billion. We do cell-based panning on the CHO lines I just showed, and we deplete at every round against the parental CHO background line. Typically, we take about 2 weeks to do that, and we do about 4 to 5 rounds of panning. At every round, we're collecting the output to look at and do NGS to basically look for enrichment of specific sequences. And at the end, we actually pick hundreds of colonies from those final outputs and do either NGS or Sanger sequencing to get the paired heavy light chain reads. And we utilize all that information plus the NGS enrichment data to give us clues as to which sequences that we should make. Rather than mess around with fragments and trying to do phage-based bindings experiments, we basically reformat directly to IgG using all that sequence data. Because we're at Twist, and we can make large numbers of genes, we can reformat up to 100 or 200 different antibodies directly to IgG, scale up that DNA, and we go directly into the production of small amounts of IgG for doing binding assays as well as functional assays. So in a matter of 2 to 3 months, we can go from our initial target all the way to potentially a functional anti-GPCR antibody. So the one case study, the first one I'll talk about today is our case study with GLP-1 receptor. So GLP-1 receptor, we panned it, again, using our stable CHO lines and depleted against background CHO. Parental, we came up with, we saw almost, several hundred antibodies that were enriched through the course of that panning. And we chose 100 of them to make. And of those 100, 15 of them bound to GLP-1 overexpressing cells and didn't bind to the background CHO line. Shown here are 8 of them. And you can see here we ran a titration of the antibody against the GLP-1R CHO line versus the parental background CHO line. And you can see in many cases EC50s in the range of single-digit nanomolar all the way to micromolar, with, again, very specific binding to the GLP-1 overexpressed line. Again, I'm highlighting the clone named GLP-1R3 because it's an important antibody that I'll go into a lot more detail later. So when we took those antibodies that now showed specific binding to the GPCR target, we then assessed their function. So the first thing we did is we ran a cyclic AMP assay against GLP-1 receptor. And again, the ligand in this case was the GLP-1 peptide. And so shown in black is the activation curve, and it's the cyclic AMP assay with the GLP-1 peptide in the absence of any antibody. We then added a high concentration of each of these 8 antibodies I just showed in the previous slide to assess how they would affect that activation in the cyclic AMP assay. And what you can see is all 8 of those antibodies either inhibited the overall signal or even shifted the activation signal to the right in this assay. So they were either all direct antagonists of the receptor or they were negative allosteric effectors. When we look more closely at these sequences that bind to GLP-1 receptor on cells, what you might expect to see is expect to see potentially some motifs that make sense based on the target you're panning on. So in this case, GLP-1 receptor, we might actually expect to see some sequences coming through in that heavy chain CDR3 loop that have the GLP-1 peptide motif, and that's exactly what we see. So what we see is all the sequences in yellow actually have the GLP-1 peptide motif embedded in the loop, which again allowed it to bind specifically to that receptor. We also saw some sequences in orange that also have the GLP-2 peptide motif, even though GLP-2 is not known to bind to GLP-1 receptor. And then we also found several fully human CDR3 sequences as well, which are shown in white. So we saw a nice variety of different sequences, and we also saw several sequences that made sense based on the target that we panned on. And the GLP-1 peptide motif was a very small percentage of the overall oligo pool, but in the context of panning against this target in phage, we were able to pull out some very specific binders that utilize this motif to guide it and bind and have function against this specific receptor. One question I'm often asked is, well, again, this motif is help guiding binding to your receptor. Is all the binding due to this motif that you've grafted into your antibody? Do the other loops in the antibody actually have any effect in that binding? So to address that question, we actually did a paratope analysis of both the heavy chain and light chain, look at the contribution of the other 5 loops in the binding of this particular GLP-1R antagonist. So what we did is we just did an alanine scan through heavy chain CDR1, 2, and also light chain CDR1, 2, and 3 to understand the contribution of each residue in each of those loops to the overall binding. And what we found is that there was some dramatic contribution for some of the other loops in the binding of these antibodies to, in this case, GLP-1 receptors. So for example, we saw that there were definitely some contributions in heavy chain CDR1 as well as in heavy chain CDR2 to the binding. And then we also found that there were contributions in light chain CDR1 and CDR3 with no contribution by light chain CDR2. So again, unlike, and to answer that question, we definitely saw a lot of contribution from the other loops, in particular, CDR1 and 2, as well as light chain CDR1 and 3 to the binding of this particular antibody to GLP-1 receptor. We then took that antibody, and again, one of the advantages of using synthetic libraries is you're oftentimes not bound by tolerance, and so you can get really nice species cross-reactivity. And so we looked at this antibody to see whether it had cross-reactivity with mouse and macaque as well as human, and we found that we saw really nice FAX binding to all 3 species. So this enabled us to do some of the in vivo studies that I'll show later, showing that this antibody actually has in vivo activity. We also took this antibody, the TV01-3 antibody, and also shown that it bound to human precursor cells that endogenously express the GLP-1 receptors. So again, much like the functional assays I showed before that just use the native sequence, here again, we're showing that these antibodies can bind the native sequence of GLP-1 receptor expressed at a much lower level in these precursor cells that endogenously express GLP-1 receptor. So we've now gone on and taken this antibody, which again, shown on the left, inhibits the activation of GLP-1 receptor in the cyclic AMP assay. We've now done a complete titration curve in this at a specific GLP-1 peptide concentration and show we can get complete inhibition of signaling using this antibody. It is a direct antagonist of the receptor. And now we've even gone on and done some in vivo studies and a glucose tolerance study to show that as you block this receptor, the mice can maintain a high level of glucose once injected with glucose. So the antibody actually can block the receptor and block the action of insulin and GLP-1 peptide secretion to actually lower the glucose levels. That we actually see a maintenance of high levels of glucose in this study. And we've even gone on and shown that these antibodies also have a very long half-life in rats with the overall T-beta half-life of over a week. So again, a really nice story to show that we were able to find a very specific functional antibody to this specific GPCR. It has a very long half-life in rats and also has function in glucose tolerance tests. So that was GLP-1 receptor. We've gone on and done POCs against a number of other GPCR targets. CXCR5 is one of them. I'm just showing this one slide showing some data from the CXCR5 study we've done. We've come up with a panel of different CXCR5 antibodies, 2 of which are shown here. Several of these antibodies are weak antagonists in a cyclic AMP assay, but we have shown that several of these antibodies have a pretty dramatic effect in a beta-arrestin assay, which is shown at the bottom of this slide. And so we're, again, continuing to follow up on these antibodies and actually optimize them for greater potency to see if we can shift much of the functional activity we're seeing, particularly in the beta-arrestin assay. We've also gone on and panned our library against CRTH2, which is, again, a target that's involved in asthma. And we found a whole panel of different antibodies that bind to our CHO lines that overexpress CRTH2 compared to the background CHO lines. So shown here in green are some of the candidates that we have found against CRTH2. Here's a couple more. And we've now taken these antibodies forward, shown that many of them, again, at high concentrations, so show a shifting in the cyclic AMP assay. And so they are either potential antagonists or negative allosteric effectors of CRTH2. And we've now gone on and optimized these antibodies further to make them more potent. And we've just now gotten some of those approved variants just this week. And so we'll be able to follow up and show that not only do they bind CRTH2 better on cells, but also potentially have greater function in this cyclic AMP assay. So just to conclude the presentation and the case studies that I just showed today, this GPCR library is performing very well against the panel of Class A and Class B GPCR targets. The antibodies derived from this library are well behaved and could potentially be drug candidates. I didn't go into all of the developability studies we've done, but we've done a whole panel of different developability assays to show that these antibodies express well, behave well, they're monomeric, they're not sticky. But again, I always say the best developability assay you can do is actually a PK study. And I showed you data in the previous slides that these antibodies do have long PK. And then for the GLP-1 antibodies, we've also shown that these antibodies, not only do they have function against GLP-1 receptor, but also bind their target on primary cells. So the overall conclusion from our POC studies to date is this motif-guided library approach is a robust path to finding functional GPCR antibodies. We have found that that's a little bit more fruitful against targets that bind a known protein ligand, like for example, the GLP-1 receptor example that we just did. But it doesn't mean that we haven't been successful against other targets that don't necessarily bind a proteinaceous ligand. And that's the conclusion of my talk today. Again, we have some time, and so I'd be happy to answer any questions you might have about the presentation I just gave. We have one question from the panel. So do these antibodies bind discontinuous epitopes? Do they detect receptor in Western blot? We haven't done a epitope analysis on the GPCRs themselves, so I can't answer the question whether they bind a discontinuous epitope. And I haven't done any Western blot experiments. All of the antibodies that we typically find bind the native GPCR. So in a Western blot, they may not work because the Western potentially would denature the GPCR structure.
Thank you, Dr. Sato. We do have a few questions that came in a bit earlier, too. The first one, have you done any assessment of the immunogenicity of the antibodies derived from your GPCR library?
That's a great question. You might again expect, since we're grafting in a non-antibody sequence into our heavy chain CDR3 loop, that could potentially introduce some immunogenicity. We have done some in silico assessment of immunogenicity, and in some cases there are some unique junctions between the antibody and the motif that will require some additional engineering. So in some cases, we will potentially have to optimize those antibodies further to potentially remove some T-cell epitopes that might be created.
Thank you. A second question we have, are the anti-GPCR antibodies stable and developable compared to other human therapeutic antibodies?
Yeah, I mentioned that just briefly before, but yeah, we've the antibodies that would come out of the library. The library itself was actually heat tempered as we built it, so it's the antibodies that come out are very stable. They behave just like any other human antibody therapeutic, and so I believe that they're potentially very great drug candidates derived from the library. We have another question from the webinar. So have you tried panning using tumor cell lines instead of stable cell lines? I have used tumor cell lines in concert with some stable cell lines. I've also tried doing panning using solubilized GPCRs as well, and I have gotten some hits using those approaches, particularly for the solubilized GPCR. I haven't done a huge amount of pannings against the tumor cell line, but I don't see why we wouldn't be able to do that as well. Again, we would just need to make sure that we're directing the library against that specific target you're going after, rather than just all of the GPCR targets that are on that tumor cell line.
Thank you. And we do have one other question that came in earlier. Have you found any agonist antibodies from your POC projects with this library?
Yeah, so when we built the library, I actually thought that we might just get a lot of agonists, because the motifs we're using are actually derived from mostly agonists against these GPCR targets. What we're finding is we're getting actually mostly antagonists. Agonists in general are really difficult to find, and so we're finding that it's also been challenging to find agonists from our library as well. And we're still trying to push the limits of what we can pull out of the library and see if we potentially can find agonists against different GPCR targets.
All right, and I just forwarded you a question from Megan.
Oh, great. From the selected clones post-screening, what percentage of them show functionality versus percentage of work in binding assay? again, when we make our antibodies, we typically just use the sequence directly to choose a large panel of sequences to make. And so from that, we typically make about 100 antibodies, and from that, maybe 15 to 20 of them show good binding in flow, so they show binding. And then of those, typically 25% to maybe 50% of those antibodies show some function in an assay. So again, you can do the math, but it ends up, after all that work, being a handful of antibodies that have good binding as well as function.
All right, that appears to be all of the questions that have come in to me. I'd like to thank you so very much, Dr. Aaron K. Sato, PhD, from Twist Biopharma, for your presentation today. I'd like to thank Twist Biopharma, a division of Twist Bioscience, for sponsoring today. And most of all, I'd like to thank those of you who came and spent this time with us. We're very grateful you could come. We know you're very busy and your time is precious. So thank you again for attending today. I look forward to seeing you at future Cambridge Health Tech Institute Global Web Symposia. Thank you all so very much, and have a great day.